Okay, welcome to this afternoon session on the interplay of AI and neuroscience. I'm Rainer GOEBEL, I'm the chair of the session, and we will have three speakers today going more from implementation up to more information processing in the kind of sequence of these three talks. And the first speaker I'm happy to welcome is Charlotte FRENKEL from the Delft University of Technology in the Netherlands. And unfortunately, due to travel issues, Charlotte could not come in person here to Marseille, but she is present online, and she will be coming soon, I guess. And let's hope that this works. Charlotte studied in Louvain, Belgium, where she got her PhD, and she did then a postdoc at the ETH in Zurich before she moved to Delft. And despite her young age, Charlotte is already a leading expert in the field of neural networks and neuromorphic engineering, as we will see in a moment. And she was awarded her PhD, which is only 2 years ago. She was awarded by several prizes from the Belgian funding agencies, including the IBM Innovation Award and the Nokia Bell Labs Scientific Award 2021 for the best PhD thesis in the field of ICT. And she will talk today about merging insights from artificial and biological neural networks for neuromorphic edge intelligence. I hope Charlotte is here. If you are here, I give you the floor. Yeah.
And for the invitation as well. It's really my pleasure to talk about how to merge insights from artificial and biological neural networks for neuromorphic intelligence. So in this talk, more specifically, we'll answer 3 questions. The first one is how to make the best of neuroscience and AI, and more specifically, what is the right perspective and what is a good starting point. The second question that we'll look at is how can neuroscience help achieve these goals? And the third one is how can we move the 2 previous questions into interesting engineering solutions? So let's start with the first one, and more specifically from the perspective point of view. So we have this tight interplay between neuroscience and AI algorithms, and there are really 2 ways to go. So first is from neuroscience to AI. We have that this is really about science. This is about uncovering the brain computational primitives that are going to be helpful for a given use case, and how to scale up and deploy these brain computational primitives into real-world AI workloads. So this is really a bottom-up approach. So for example, there's the field of deploying STDP for neural network training. This is really this bottom-up approach. But on the other hand, this is more about engineering. You can also start from existing AI algorithms and modify them so that they can align with neuroscience insight. So for example, this is what the whole field of bioplausible error backpropagation is about. So these are really these 2 complementary approaches. But the problem is that we lack a perspective as to how to make the best on both sides. Which one is the most promising is still a bit unclear. And so the standpoint that I'll take in this talk is that hardware forms a good middle ground in between. So why? First, there is an obvious synergy between AI and hardware, because AI without hardware is unsustainable. I guess most of you are already familiar with this kind of graph showing the evolution of training compute over time. For example, here in blue with the conventional machine learning workloads, and now since the transformer era, this is completely unsustainable to continue scaling up the GPU server farms. At some point we'll hit an economic constraint. And so we need dedicated hardware to sustain this huge growth. But on the other hand, usually the synergies between hardware and neuroscience are a little bit less talked about. So this is going to be the focus of the second part of this talk. How can neuroscience help, and what is the interplay with hardware? So let's explore both sides. First, from neuroscience to AI, so the bottom-up approach. I'll focus here on synaptic plasticity, and we'll see why shortly. So for now, the starting point is synaptic plasticity. And usually, starting with this, the main learning law that everybody talks about is spike time independent plasticity, also the oldest and most well-known one, so STDP for short. So STDP is just about spike timing, and I guess also most of you know it. So when the presynaptic spike happens before the postsynaptic spike, the relationship is causal and you will potentiate your synaptic weight. Otherwise, it is anticausal and you depress the synaptic weight. So this is a local learning rule that takes place in between 2 neurons. There's another learning rule that is called spike-dependent synaptic plasticity, or DSP for short. I'm not going to talk about how exactly this learning rule works. You just have to remember the basic mechanism, which is when there's a presynaptic spike, you look up the state of the postsynaptic neuron at the time of the presynaptic spike. So the mechanism itself is slightly different, but these two learning rules enforce correlation and usually lead to the same kind of learning performance. So how should we choose between both? So I mentioned before, hardware is an interesting middle ground. So let's see what it translates to in terms of hardware requirements. So on the left, this is a digital implementation of an STDP synapse. And you can see here that this is a shift register whose only purpose is to generate a time window that is able to capture the time dependency between a presynaptic spike and a postsynaptic spike. And the big problem is that you have to replicate that for every single synapse in the network. So this has a huge cost, which you do not have for the DSP synapse. Because when you get a presynaptic spike, you just look up the state of the postsynaptic neuron. So that means that the key computation is done at the neuron level and can be amortized over the whole bunch of synapses that are incoming to that neuron. So this one is much cheaper. And why is it so? Now we can come back to the learning rule. It is because these two learning rules are local, but it's not the end of the story. They are both local in space, in between two neighboring neurons, but they are not local in time, at least for STDP. This is not local in time. And this is why you need to implement the time window generation in hardware, and that is expensive, which you do not have to do in SDSP, which is local in time. Just look up the state of the postsynaptic neuron at the time of the presynaptic spike. So this not only hints to more efficient hardware, but also to more bioplausible learning rules. So for example, from this paper, you can see that voltage-based learning rules are much closer to the biophysics. And regarding the hardware savings, this is, I'm not going to talk for the sake of time about this today, but this is in this paper. This really leads to huge savings in silicon, enforcing space and time locality. So that's about the neuroscience to AI, but what about the AI to neuroscience direction? So here we start from well-known algorithms, and usually the best starting point on this side is the error backpropagation algorithm. So now bioplausibility is the end goal. So we'll first identify what are the key issues of backprop to make it more bioplausible, and then we'll release those. So one key issue is known as the weight transport. So this is just about using the transpose of the weight matrix in the backward path compared to the forward path. So what that means is that in the brain, this would imply bidirectional synapses, which makes no sense. So this is biologically implausible. And for those of you who are familiar with feedback alignment algorithms solve the weight transport problem by replacing this matrix by a fixed random connectivity matrix to release the weight transport. The second issue is update locking. So for example, if you look at this weight matrix, you cannot update it before you do the full forward pass followed by the full backward pass. So that's known as update locking. You have to wait for this two-phase forward-backward. And this is also biologically implausible because the brain has no such two phases for learning. Everything happens in parallel and in real time. So the problem of update locking is that there's no way to release this as long as you do not get locally a proxy for the error, because otherwise you always have to wait until you get the error at the end. So to solve both issues, we have proposed an algorithm that is known as direct random target projection, which not only uses fixed random connectivity matrices to release the weight transport, as you can see here, but also for classification problems, uses the target vector for classification, which is just a classification label. And we show that this can act as a good proxy for the sign of the error. So given that this information is now known in advance, we do not need to wait for it, and therefore that releases update locking. So this is one way to go to release the two issues there. But what I would like to mention is that by doing so, and then we look into neuroscience, we can see that this also aligns with some recent neuroscience insight. And more specifically in cortical intermediate layer neurons, we have some output-independent target signals. And this is exactly the case here. The target signals come directly from the input and are output-independent. So this not only better aligns with neuroscience, but by doing so, we have also reduced the computational and the memory cost up to the point that now we can implement this direct random target projection learning rule into silicon. And we can show that this is a low-cost approach because it leads to only about 15% overhead in power and area compared to the inference-only network. So this is all for the neuroscience to AI or AI to neuroscience. But what I would like to mention and what I would like you to remember is that all of this is for this claim. Hardware efficiently and bioplausibility are often two sides of the same coin. With the first approach, we have seen that designing efficient hardware hints towards bioplausible mechanisms. And with the second one, we have seen that bringing AI closer to neuroscience leads to hardware efficiency. So knowing this, now we can go back to the big picture and see that actually there's a quite nice natural synergy between neuroscience and hardware. So now how can we go about the starting point? Because we have still not looked at this question, only about the perspective which is brought by hardware. So now we need the methodology to make the best of both worlds. So first we have investigated the science-driven approach, which is fully bottom-up. And this is great for analysis by synthesis and understanding by building. But this is knowingly difficult to scale efficiently to real-world problems. So for example, these STDP trained neural networks are typically really hard to get to acceptable performance levels. The second one is the engineering approach, which is top-down and doesn't have this problem because it really starts from working solutions to real-world problems. But the open question then is, what kind of salt and pepper do we need from neuroscience in order to give a boost to these real-world solutions and lead to a real competitive advantage? And so yet another standpoint that I'll take in this talk is that to make the best of both worlds, which we denote as neuromorphic intelligence, we should use the second approach fed by insight from the first one. And so now let's go to the last part of the talk. How can we morph these questions, and more specifically this methodology, into interesting engineering solutions? So interesting engineering solutions means first selecting a use case. And for this use case, now you understand why I focused previously on synaptic plasticity and neural network training algorithms, is because the use case that I look at is the one of on-chip learning. So why so? We have that for every inference device, as long as the input data is within the distribution that was used during training, the performance is going to be within the specifications. But as soon as you have out of distribution data, the performance is going to drop below the specifications. So how can we deal with that? Because the problem is that it comes from different users, different environments, and also task requirements that can also evolve over time. So at release time, you may be within the specifications, but over time, your specifications might actually degrade. So one way to go is to use more training data before deployment, but as I just mentioned, this is never a guarantee of robustness and flexibility in the long term, and this has a cost of gathering more data. The second one is that you can exchange data with the cloud, but you might not want to leak user data to the cloud, and this comes at a power budget increase. So this is why we'll look at on-chip training, and more specifically, we'll make it bio-inspired and end-to-end. And just to clarify, end-to-end means here no external memory, because external memory is what is costly in hardware. Accessing this memory costs a lot of power. But the problem is that for video and speech data, so any kind of temporal data, deploying on-chip training end-to-end without external memory has not been achieved to date. Why so? Because the workhorse of deep learning is the backpropagation through time algorithm when it comes to temporal data. So for example, let's assume here you have temporal data. So you have a 1 second sample at a millisecond temporal resolutions. That means 1,000 time steps. And for each of these time steps, you have to keep a copy of the network state at each time step so that when you get the final error, you can backpropagate through the whole network dynamics. So that means that you need to keep a copy of all time steps, and that has a lot of memory overhead. Obviously, the brain doesn't do that. So another family of training algorithms is known as forward mode training algorithms, among which the eligibility propagation, or EPROP for short. So EPROP works by just approximating the backprop through time gradients as the product of two terms. One that is a learning signal, that is an error-based dependent term here, that is approximated by EPROP to be available locally in time. So that's the main assumption of EPROP. The second term is an eligibility trace. So eligibility traces are purely forward terms. They are not error dependent, and they just capture basically the importance of every synapse onto the network output. So they are computed exclusively forward. And so that allows you basically to keep only the current time step in memory. So that's much cheaper. And going from one to the other, you not only increase biological plausibility because synaptic eligibility traces are supported by biophysical evidence, you increase space and time locality, which we have seen is the magic recipe, and therefore you slash the on-chip memory requirements. So just a quick note here, EPROP should be amenable to a hardware implementation thanks to space and time locality. But to enforce this, you need to modify a little bit the eligibility trace formulation, which I won't do in this talk. So here you'll have to believe me for now. But what I would like to focus on is the methodology. So I told you that neuromorphic intelligence 2 should be fed by 1. So let's apply this. We start from backpropagation through time, the workhorse of deep learning. But the problem is that it's not tractable from a hardware point of view, it's not bioplausible. So we have looked into neuroscience. Neuroscience says, okay, a legibility trace might be the good way to go. The combination of both is the EPROP algorithm. But EPROP per se cannot deal with the long timescales without a good neuron model that embeds long temporal dynamics, and is not hardware efficient without space and time locality. So looking into neuroscience, the magic recipe on the neuron side is to embed firing threshold adaptation over hundreds of milliseconds. But this leads to an EPROP formulation that is intractable in hardware. So we leave neuroscience and we go back to engineering, and we show that using a completely standard leaky integrin fire neuron, but whose membrane potential leakage time constant can be increased to seconds, this is not bioplausible, but this solves the problem. You combine both with space and time locality, which is a bit of rework, and you end up with simplified EPROP. So you can implement that directly on chip. This is a Reckon chip. This is also fully open source. And it's a low-cost solution, only 0.5 millimeter squared and no external memory. It implements a spiking recurrent neural network topology, and everything is spiking, including in the input, which allows it to be event-driven and sparsity aware, but also to be sensor agnostic and be able to directly process the raw data from the sensor as long as the sensor talks with spikes, for example, silicon retina, silicon cochlea. So what is really nice with this property is that it allows, therefore, for task-agnostic processing and learning. And to demonstrate this, we took 3 benchmarks, one on vision, one on audition, and one for navigation. So gesture recognition, keyword spotting, and this usual neuroscience task of the rodent moving along a corridor and having to decide to turn left or right. And all of these tasks last for about from 1 to 6 seconds and embed short timescale temporal dynamics on the 1 to 5 millisecond timescale. And yet Reckon was able to learn all of these tasks from scratch without external memory with a very good performance, as you can see here, reasonable accuracy levels. And all of this with a power consumption that is below 50 microwatts at 0.5 volts. So this is really the kind of power consumption that is suitable for wearables, for example. So what you have to remember is that Reckon merges AI, neuroscience, and hardware to achieve end-to-end on-chip learning over second-long timescales while keeping a millisecond temporal resolution, which is a yet unsolved engineering challenge. The second one is that it provides a low-cost solution, less than 0.5 millimeter squared of silicon area, and less than 50 microwatts for training. And the last one is that it demonstrates task-agnostic learning with a spike-based encoding towards user customization and even full chip repurposing at the edge. So that's linked to electronic wasting, indeed. And so this outlines an exciting future for neuromorphic edge intelligence. And so that's a good opportunity for me to thank my former collaborators. And given that my team is now being assembled in Delft, I should have some positions open soon. So if you're interested, just drop me an email. And yeah, that's all for me. Here are some references and the ways to contact me. Thank you very much for your attention.
Thank you very much, Charlotte, for this wonderful talk. Very insightful, very exciting. And we have decided, because of time, to have questions at the end of all 3 talks. Otherwise, it takes time to go to the microphones and so on. So we give you the opportunity of questions, but after the next 2 talks. And Charlotte will be then back. So it's my great pleasure now to introduce Mihai PETROVICI. He's a leading expert in the field of bio-inspired AI and neuromorphic computing. And he studied physics in Heidelberg, where he worked with the great Karl-Heinz Meyer, which of course HPP owes a lot. Mihai leads the NeuroTMA lab in Bern and co-leads the computational neuroscience group with Walter Senn. Mihai and his team impress me all the time by being super productive. They publish a stream of highly influential papers in very high impactful journals. And I hope you will get a glimpse of this kind of super exciting work now when Mihai talks about computing with physics, learning from carbon and silicon. Mihai, the floor is yours.
All right, thank you very much, Rainer, for this very kind introduction. And thanks a lot, Charlotte, for your wonderful talk. I hope you can hear me. I'm looking at the screen instead of looking at you. I think you will find that I'm going to echo quite a few of the sentiments that you have expressed in your talk. So we're interested in computing with physical systems. And of course, I would say the archetype of a physical system that is really good at computation is the brain, so we have a lot to learn from there. But we're also interested in building physical systems that are powerful computers and hopefully one day will rival the abilities of the human brain. So what does it take to build a physical system? And let's go for a really big challenge, something like a universe. This is the Millennium simulation. It's actually a really old video from 20 years ago. And what they succeeded here to do is to really simulate a huge chunk of the observable universe. An important reason why they were able to do that is that we have a very precise understanding of the fundamental interaction between components at that scale, and that is gravity. Now, if you want to do the same thing for the brain, it's significantly more difficult. And the reason is that we don't yet have a good understanding of the microscopic dynamics that are essential for computation in the brain. So some of you might remember this slide that I have shown 5 years ago at the Science Alignment meeting in Alpbach, where I expressed the same sentiment and I outlined a little bit of a scientific agenda for the years to come for some of the sub-projects in the HPP, and the core idea was exactly this: find the computational primitives in computing systems, from those as complex as the brain to those as relatively simple as those we use in machine learning, and what are the components that have the mechanics to implement these computations. And this has been an ongoing effort in the HPP, not even before this talk that I gave, but afterwards in some of the sub-projects. You might remember CDP5, and now in Work Package 3. So, the equations are, for those of you who have already heard me talk about these topics, as a little bit of a challenge. For everybody else, please focus on the intuition and ignore the equations for now. So, this is a very physicalist perspective, or a physicist's perspective, but I hope that when I say that the energy of a system is a good characteristic of it, I hope that this echoes also with your experience. So in physics in general, from the energy, if you know the energy of a system, you can derive lots of things. Now, what would be the energy of a computational system? Arguably, we could say that in physics, the energy is a measure of the internal frustration of a system. So for a computing system, the energy could be said to measure the wrongness, how bad it is at performing a certain task. Now, if you have a system that is shielded from the external world, then there is no right and wrong. So the energy is zero. This would be the blue trajectory over there in the plot. As soon as we let the system interact with the environment, if it hasn't learned anything, it's going to do something wrong. So the energy is going to go up. It's going to move up the mountain. It's going to be positive. And learning in the system is supposed to make the system do the right thing eventually over time. So it's driving the trajectory of the system from up there on the mountain back into the valley. And that's the green trajectory. So we can put all of this into math, into equations, and then we can apply another intuitive principle from physics, namely energy conservation, to derive dynamics. So you all know that energy is conserved, and from this you can derive the dynamics of any system in principle, including also the dynamics of neuronal systems. And what you get here is an equation that should be familiar to many of you, namely the leak, integrate, and fire equation. So you see, from simple physical principles, we rediscover deep knowledge about biology, and even more than that. The components of this equation correspond to functional components in pyramidal neurons. You can see a structural subdivision into functional units: the soma, basal dendrites, apical dendrites. So this approach also tells us something about neuronal morphology. As I said before, what are synaptic dynamics supposed to do? They're supposed to teach the system to do the right thing. So they're supposed to reduce the energy. Now, the math here is slightly more complicated, but the result points to other structures in the brain. For example, the existence of microcircuits. So the interaction between pyramidal and interneurons to calculate errors, to propagate them throughout the brain, to instantiate actually a form of biological error backpropagation, and with a couple more ingredients, to actually instantiate feedback learning that also Charlotte has talked about, so to learn connections both from lower to higher cortical areas and vice versa. One nice little ingredient in this theory that turns out in the end to not be that little at all, is the concept of prospective coding, which I guess goes a little bit against established wisdom in the field, namely that neurons don't just react to their current input, but they're actually, every individual neuron in itself, they actually try to predict what's going to happen in the future. And we actually know that this is the case even from the oldest models of neuronal dynamics. In Hodgkin and Huxley model, you can see that neurons react to the speed with which their stimulus changes. And this also corresponds to data that has been measured more recently, where pyramidal neurons actually fire in anticipation of their inputs. So, with all of these ingredients, we can not only explain structures in the brain, but we can also use them for applications. Here's an example of image classification or something more exotic, finding Higgs particles in data from CERN. And in both cases, our biologically plausible and biologically inspired models are on par with state-of-the-art approaches in machine learning. And there's a nice little detail to these models, namely that if you look at the structures that emerge in the higher cortical areas in these models, you'll see this nice delineation in their activity. You'll see how activity is nicely clustered. We call that linear separability. And this clustering, one could also call it sementization, should remind you of something, namely of sleep, because we know that sleep is there to better structure our memories. So we can use these models to actually understand things about sleep, to understand the role of different phases of sleep, for example, of NREM, where memories are consolidated, and of REM sleep, which is characterized by more creativity, as I guess everyone can attest to who has ever had more weird dreams. So we can then turn off these individual sleep phases in our models and see what goes wrong. And we can see that both sleep phases are necessary in order to achieve a nicely structured activity in higher cortical areas, such as hippocampus. Now, something that I glossed over a little bit on my previous slides, you might have noticed, is the way that information is being transmitted in both the brain and many neuromorphic systems, namely spikes. And spikes are a bit troublesome because they're discontinuous. So if you want to apply the same energy picture to a spiking network, the energy landscape is going to be jagged and rough. It's very difficult to drive trajectories down that landscape. However, it turns out that if you look a little bit closer, a lot of information is encoded not in the spike itself, but in the timing of the spike. And now, the timing of a spike, again, becomes a continuous function of the timing of the inputs to the neuron and of the synaptic weights. So suddenly we have another, again, a smooth landscape. We can calculate gradients, and what we get from the same energy-based approach is a slightly more complicated synaptic learning rule, but one that depends purely on pre- and post-synaptic spike timing. So this is, if you will, a more complicated version of STDP. And we can use it to train spiking networks. Here's an example of a network that learns to classify images. You can see how the correct neuron, the neuron with the correct color, always learns to spike first, so the image is correctly classified. And you will see quite a lot of diversity in these plots. And the reason is that this is not one of those nice, clean simulations, but actually a physical system doing the computation, namely a neuromorphic chip, in particular the BrainScales 2 chip developed in Heidelberg. And this chip has a couple of nice features, two of them being that, first of all, it's accelerated compared to to biological neural dynamics by several orders of magnitude, and it consumes very little energy. So with this chip, we can classify, we can process more than 10,000 images in 1 second with less than 10 microjoules per image. And here's, just to put this into perspective, a banana for scale. So this corresponds to 30 billion classifications on the energy budget of one banana. Now, I am aware that this was a bit fast, but you can hear more tomorrow. I'd like to recommend the Brain-Inspired Robotics and Computing session at 10:30, where Julian is going to tell you more about this approach. I implied just before that precise spike timing is important. This is certainly true in some brain areas and for certain functionalities. But we know that the brain is stochastic. So this stochasticity can be also interpreted mathematically precisely, namely that it relates to probabilities. So we are confronted very often with uncertain data. And here's an example, this bistable image of a duck and a rabbit. You don't know which of them is there. So you need to have a representation of multiple plausible realities, a probabilistic interpretation. And again, you can define an energy-based framework to describe these probabilities, and from this framework derive neuronal dynamics, and lo and behold, you get, again, dynamics that are well known to us, namely leak, integrate, and fire dynamics. And in particular, you can notice that in order to perform these kind of computations, neurons need to be in a high conductance state. So this hints towards a functional property of the high conductance state. There's another more difficult issue here, namely the ability to switch between representations. This is something that is quite difficult in machine learning, for example. If you're convinced that this is a rabbit and you don't see the duck, this might not be that much of a problem, but if it was a predator, you might end up not being able to pass on your genes. So it's really important to be able to have an open mind, to change your mind about possible compatible realities. And the math points the way towards the features of the brain that enable us to have an open mind. And here are two examples, one of them being short-term plasticity, and the other one being cortical oscillations that modulate the energy landscape in a way that allows the brain to switch between modes. And here's an example of a spiking network that was trained on images of children's toys, and without the cortical oscillations, it is stuck in a single mode, and with simulated oscillations, it can switch between its different memories. I'll just mention this in passing, but I think this is pretty cool that research sometimes has rather serendipitous outcomes. We did not plan to do this, but it turned out that this is an interesting thing to do. Namely, if you have a Bayesian framework, a way to represent probabilities exactly, you don't have to necessarily apply this to a classical system. You can also apply it to a quantum system by doing the right math again. So it turns out that we can use the neuromorphic devices that we use also for other applications to represent quantum systems and measure their properties. It's called quantum tomography. And I find this really exciting because you're using a classical system to demonstrate some deeply quantum phenomena such as superposition and entanglement. So this brings me to the end. I think it's pretty clear that all of this work has been the result of a lot of effort from a large and very diverse team of researchers. I think this is a testament to the HPP that so many groups were brought together under this umbrella and were able to collaborate, bringing together experts across many different fields, computational neuroscience, machine learning, neuromorphics. Unfortunately, I do not have the time to mention every one of these great researchers by name, but I will mention one name whose drive has been with us for a long time, without whom we could not have done this work, and whose passion and dedication was fundamental for our project, and with whom we probably would not be here today, and that name is Karl-Heinz Meyer. Thank you.
Thanks so much, Mihai, for this fascinating talk. And hope we will have time for questions, although I see I have a little bit of a conflict. Time is running short, but I also want to present at least a bit of my talk. Can I have my slides? I don't see them there. Perfect. Yeah, what I want to do is going a bit in more higher level computational and cognitive neuroscience, because in the work we are doing, we are interested in basically going from the brain-derived modeling often embodied modeling, but then also learn new insights, which you want to bring back into our brain knowledge, and hopefully even in a testable way. That's my main interest in this work, to really learn from these often embodied models. So here's an example of an architecture which was built by a team mainly based in my university in Maastricht, and actually headed by Mario Senten, who could not be here, unfortunately, because of travel issues. The idea here is that we build modular architectures, which often quite heterogeneous modules. Some are kind of AI-like, like deep neural nets. Others are more spiking networks with handcrafted connections, and others are inspired by simple neural mass models. The point is, what I want to highlight today is I'm focusing on two components of one of these models we built. This is for Sackett's for scene understanding, and I want to focus on two structures where this idea to go from the brain to the models and then back into the brain and testing hypotheses which were generated by the models, I want to give you two examples. The first is on what we call retinal sampling, and explain this in a moment, and the second will be on saliency detection. And actually, for the second work, Alexander Kroner did the major work here, and he's here at the conference. So if you have questions to that, detailed questions, he can answer you all the details. But I will start with a general explanation what I mean here. And it goes back to an interesting paper by Yammins and Di Carlo some years ago. The main idea is that we hope when we do brain-derived models, and we then add to these brain-derived models the powerful learning of AI and good representative huge data sets, then we have a chance that these AI models pick up computations by finding weights to solve the task, which might help us to inspire our research how the brain solves the same task. And of course, that is not necessary that the solutions are similar. But if we do brain-derived research, as we do in the HPP, the chance is higher that what these AI learning rules find can help us to understand computations, and that is the productive part. Often we can derive testable new hypotheses, and then we have a nice modeling empirical closed loop. And I want to show you this in two examples where I think this worked already quite nicely. So the first example, as I said, is on retinal sampling. And the idea here is that all classical AI models, convolutional deep networks in vision, I focus on vision only today, and in these networks, we basically feed the networks with photographs with a kind of equal pixel sizes, regularly spaced, right? This is what is the input layer into these networks. Usually 3 of those layers, 2 for space, 1 for RGB. And this is the input in classical deep learning networks, convolutional networks. If we look in the human brain, there's a striking difference, which is shown here in the middle. If you see this picture here, the retinal ganglion cell density image, then you see that this is actually how an image looks at the retina, and that means it's the input via the LGN to the primary visual cortex. And for example, if you have an image and focus here on the middle point, which is what's happening here, then you see that we have a representation at the retina which enhances massively the foveal part, and it shrinks the peripheral part, which gets lower resolved and also shrunken because we have less density sampling ganglion cells in the periphery. So this is a very simple change. But we ask the question, what happens if we use or extend classical deep learning networks with such a retinal sampling so that the input images in a classical convolutional network is like it is in the human brain? Very simple. And just to stress, somehow our consciousness lives on a kind of re-established image. It's somehow in the pipeline is an inverse mapping. What we experience is, of course, not a representation like shown here in the middle. And of course, that's also an interesting question to understand. So the point I want to make is the following, and this is work by Danny da Costa and our team. The idea is very simple. We make from the foveal to the peripheral part on the input, we basically keep a constant filter size. But what is here indicated by this enlargement is that the ganglion density increases. So that means that we automatically get larger receptor fields in the periphery, and we get also sensitivity for lower spatial frequencies in the periphery as compared to the foveal vision. This falls out naturally from this assumption. The other point is when we apply not the standard regular image, spaced image, but we use this kind of realistic ganglion input to these neural networks, then you see this is a so-called barrel distortion. That means if I'm having my focus here in the middle and have a radial edge like this line here, I detect it better than I have here kind of this edge, but I have kind of this orientation, because then the orthogonal edge appears curved here. And the filter here gets weaker activity. It sees the edge, but less good. And actually, a similar phenomenon is known from the human. It's called a radial bias. And we will see that this effort we do here, among other things, leads to a new hypothesis, which not only what causes the radial bias, but also that it's spatial frequency dependent, which is not yet known and has to be validated. So the point is now here, what we did is we used a biological standard convolutional deep network, the CoreNet-Z model, which is from the DiCarlo and Yamins group. And this model is already biological in the sense that the progress in convolutional networks moves away from the original biologically inspired ones. So you find these days networks with slightly better performance than this network and more older networks, but with hundreds of layers and architectures which are no longer easily linkable back to the brain. And these authors made the CoreNet-C model and other variants of the CoreNet model precisely to be close to the brain. So it's a biologically inspired but AI deep neural network. And the only change we did to that is we had as input not the standard rectangular pixel image, but we had the output of our simulation of the retinal ganglion sampling as input, the images you saw before, distorted images. These we feed now as input. And how do you test what the effect of retinal sampling is in the brain? Things like cortical magnification, of eccentricity, polar angle dependence, and several kind of classical organizational principles we are well known in vision research. Here shows you just how, roughly speaking, if you focus where the red point is, you would see in the right hemisphere the things on the left side, but distorted in the way you see here. And this is a signal of cortical magnification because we have more cells in the foveal vision. And the point is, when we use these modified networks, we want to quantify that, and we use the same techniques we use in the human brain on our convolutional neural nets. So we use PRF mapping or retinotopic mapping in a certain way, population receptor field mapping, and apply this technique, which you see here on a human brain. We apply the same technique with some modifications to our extended network. And what you see is that the classical model with standard pixel images shows, for example, no dependency on eccentricity here in V1, V2, V4. These are the models in the core set. There's also IT, but these are the three early layers of this core set network. And you find here in our modified version behavior which looks very similar like in the human brain. You find cortical magnification, you find eccentricity gradients, and polar angle as as it is more in the human brain. More strikingly, you see this here on the size of receptor fields, where you see there is not much dependence on eccentricity in the standard networks, but there are increasing receptor fields from fovea to peripheral vision, but also across the visual areas, fitting very qualitatively extremely well what we know from the human brain. So with this very slight modification, we get these benefits. And now I come to the striking new point. If we apply this for the so-called radial bias, the system replicates for high-frequency spatial frequencies, exactly the findings, even of course nicer, more clear than found empirically in the human brain. But the interesting point is if we go to low spatial frequencies, the effect reverses. So suddenly in low spatial frequencies, we not see the radial edges best, but orthogonal to that, which was very surprising in our modeling. And now we did a first imaging analysis, this is very early work, and found evidence also in the human brain. So if you look here, you find that for 45 degrees, we find kind of an inverted effect if we lower the spatial frequency. So that has to be continued, but it shows that the hypothesis from the convolutional networks that the radial bias is frequency dependent could resolve a still ongoing debate of the cause of the radial bias. Now I want to switch to the second example, and this is saliency detection. And what we here try to do is to see how human saliency works, what information is used, for example, to make the first eye movement, to what do we look, what do we find interesting in the environment. And the classical networks of saliency use mainly contrasts of features, feature maps. And in the classical, for example, well-known Itikorren-Niebuhr model, you see here how it works. You have here an input, then you analyze it, for example, for contrasts in color, in orientation filters, in spatial frequency, and so on. And you find differences there, and then you accumulate them in a saliency map, which gives you then input to saccadic eye movements, right, or attention, spatial attention. And the point is, these work well in lab stimuli and some of these naturalistic stimuli, but they fail also in many cases. And what we set up, especially Alexander Kroner, was the idea to use, again, a standard deep learning network, but now a object recognition one trained on ImageNet database, but now cut the upper layers and add a decoding network and train this for input natural images with from human databases, large databases, where humans look at by eye movements or by touching on the screen what they find interesting, to have kind of these calculated saliency maps from thousands of examples from where humans look at. If we train this network, which has some other specialities which I cannot talk about time reasons, we can imagine that because it's a ventral stream in the sense of its object recognition, we add, so to say, higher level object semantics to saliency. This is the difference to the classical models. And what is the impact of that? It's quite striking because when you have new images to test this trained network, you find that here, ground truth, this is the human data, that the network is scarily close to what humans do. So it predicts for new data, natural images, almost perfectly where humans would look at, where they would use the first saccade and the next one, so the most salient stimuli. And even more striking is when we compare what humans do to our model. Also here it's very similar, but look what happens in comparison to the classical models. It deviates quite strongly because the classical models focus on low-level contrast. And you see here that they focus, for example, here where the face is, not at the face, but here at the contrast in these lines in the background. Whereas both the humans and our model and similar other models focus here on the face. And that's quite striking because it makes a new hypothesis, namely that for saliency, we not only need the parietal stream of feature contrast and the saliency map in the parietal cortex, but we make the prediction that we also need the ventral stream, as you just saw, because that adds and explains a lot of human data. And that's new, this hypothesis that we need the ventral stream to contribute to saliency. And this is what we now tested using 7T data. Actually, we wanted to do this ourselves, but then we found a database, the so-called natural scenes dataset, which basically has a lot of 7T scans where 8 subjects were so brave to view thousands of natural scenes in, I think, 20 to 30 scanning sessions. So these subjects see the same images as the networks, and we now can compare kind of the network state, kind of the saliency maps, which parts of the human brain predict and simulate and correspond to these kind of saliency calculations from the model. And what we found is if we use in red, you see models like the Itty-Koch model, which focus on lower level complexity. And in blue, you see the models like our developed model. And you see that the early visual areas do not differentiate at all. But if you go to mid-ventral areas, this is mainly area V4 in the human brain, but also mid-parietal, and then in parietal and ventral, you see that there is a much better explanation or more similarity to the saliency calculated by our model than in the simple models in red. And this also shows that we can find now evidence and compare directly predictions coming from deep neural networks with empirical data measured, for example, in this case, on 7 Tesla fMRI. And with that, I want to conclude. I hope that you are convinced that these exercises demonstrates the usefulness to exploit the power of AI learning for computational and cognitive neuroscience, because it generates testable predictions which we can indeed really empirically validate. And it's clear that the AI power lies in the fact that they can develop and find weights much faster and better than humans can do, because all the traditional models, including a model I did in my PhD, were largely handcrafted. And I think it's clear that this we shouldn't do. We should let these networks, these kind of AI learning tools do this for us, or biological plausible learning is of course also possible. But the point I want to make, the likelihood increases by using brain-derived models, because the more similar the architecture and the connection patterns are to what we know from the brain, plus good databases of human data, which kind of implement the task of examples, this together forces these tools to develop solutions which are hopefully close to what the brain also found as solutions. And with that, I thank you very much for your attention.